Good morning. Today I am going to talk about one of the major macromolecules, that is proteins. Proteins are the most versatile macromolecules in living systems and serve crucial functions in essentially all biological processes. As you know that proteins are part of hair and nail. In the blood we have hemoglobin. In the brain we have got some channel proteins which are very important for us. All or most of the enzymes are proteinaceous in nature. In the muscles we have actin and myosin, and antibodies, which are an important part of the immune system, are composed of glycoproteins. So proteins serve diverse functions related to structure. They are structural components of cell. They are motor proteins, enzymes, antibodies, hormones, and transport proteins like hemoglobin and myoglobin. All proteins are polymers of amino acids, with each amino acid residue joined to its neighbor by a specific type of covalent bond. 20 different amino acids are commonly found in proteins. All the 20 amino acids are alpha amino acids. They have a carboxyl group and an amino group bonded to the same carbon atom. So they differ from each other only in their side chains or R groups, which vary in structure, size, and electric charge, and which influence the solubility of the amino acid in water. The common amino acids of proteins have been assigned 3-letter abbreviations and 1-letter symbol, except for asparagine, glutamine, isoleucine, and tryptophan. For all common amino acids except glycine, the carbon is bonded to 4 different groups: a carboxyl group, an amino group, an R group, and hydrogen atom. The alpha carbon atom is thus a chiral center, and thus amino acids have 2 possible stereoisomers. Since they are non-superimposable mirror images of each other, the 2 forms represent a class of stereoisomers called enantiomers. Amino acids so have an amino group as well as a carboxyl group. So they are known as dipolars, positive charges because of NH3+ and negative charges because of COO-. All molecules that have a chiral center are also optically active. That is, they can rotate the plane of polarized light either towards the right or towards the left. For all chiral compounds, stereoisomers having a configuration related to that of L are generally seen in the biological macromolecules. Chirality of amino acid is like that we have seen in the case of glyceraldehyde. If the hydroxyl group is on the right hand side, it is D-glyceraldehyde. If hydroxyl group is on the left hand side, it is L. Same way, if NH3+ is on the right hand side, it is D amino acid. If it is on the left hand side, it is L amino acid. Nearly all biological compounds with a chiral center occur naturally in only one stereoisomer form, that is either D or L. The amino acid residue in protein molecules are exclusively L. D-amino residue have been found only in a few generally small peptides, including some peptides of bacterial cell wall and certain antibiotics. The classification of amino acid is based on the side chain, that is the R group. So 20 kinds of side chains varying in size, shape, charge, hydrogen bonding capacity, hydrophobic character, and chemical reactivity are commonly found in proteins. So first category is non-polar aliphatic or aromatic R group. The second category is polar uncharged R group, which includes serine and threonine. The third category is polar uncharged once again, in which we have cysteine, which has got sulfhydryl group. Then we have got amino acid which have got positive charge and they are polar. These amino acids are known as basic amino acid, and the example is lysine, arginine, and histidine. The amino acid with side chain carboxylate and carboxamides. This include asparagine, glutamine, aspartic acid, and glutamic acid. In addition to these, there are some uncommon amino acids as well. That is 4-hydroxyproline, 5-hydroxylysine, methyl lysine, gamma carboxyglutamate, desmosine, cysteine, ornithine, and citrulline. So molecules, amino acids, are also called as zwitterions. They become zwitterions when you dissolve them in aqueous solution at about neutral pH. So molecules that contain an equal number of ionizable group of opposite charges and therefore bear no net charge are termed zwitterions. Amino acids in solution at neutral pH exist predominantly as dipolar ions, also called zwitter ions. In the dipolar form, the amino group is protonated and the carboxyl group is deprotonated. The ionization state of an amino acid varies with the pH. A zwitter ion can act either as an acid or as a base, that is, as a proton donor or a proton acceptor. Substances having this dual nature are called amphoteric, are also known as ampholites. Ionization of amino acids. In acid solution, that is pH 1, the amino group is protonated and the carboxyl group is not dissociated. As the pH is raised, the carboxylic acid is the first group to give up a proton. The dipolar form persists until the pH approaches 9, when the protonated amino group loses a proton. As in the graph, we can see that zwitter ion is being formed and H plus is continuously lost as we raise the pH of the solution. So, as seen in the graph, we can say that amino acids have characteristic titration curve. Now, what is the information given by this titration curve? First, it gives a quantitative measure of the pKa of each of the two ionizing group, 2.34 for COH group and 9.60 for the NH3 plus group. The second piece of information provided by the titration curve is that this amino acid has two regions of buffering power. As you can see clearly in the graph, the buffering capacity is seen around the pK value 2.3 and the pK value 9.69. Now if you see the point of inflection, that is known as the isoelectric point, and it is also called as the isoelectric pH. How will we define this? Isoelectric pH is the pH at which majority of molecules of a compound in solution have no net charge. For example, Pi for glycine is midway between the pKa and pKb values, and we can calculate the Pi values if we know pKa and pKb value. Another piece of information derived from the titration curve of amino acid is the relationship between its net electric charge and the pH of the solution. The characteristic pH at which the net electric charge is 0 is called the isoelectric point or pH, or it is also designated as Pi. Now about the peptide bond. Proteins are linear polymers formed by linking alpha carboxyl group of one amino acid to alpha amino group of another amino acid with a peptide bond. It is also called amide bond. The formation of dipeptide from two amino acid is accompanied by loss of water molecule. Peptide bond formation is therefore an example of condensation reaction, which is a common class of reaction in living system. As you can see, with the condensation of two peptides, there is loss of water molecule. And in the next slide also we can see with the red dot the formation of the peptide bond. A polypeptide chain has a polarity since an amino group at one end and carboxyl group at the other. By convention, the amino end is taken to be the beginning of the polypeptide chain, and so the sequence of amino acid in a polypeptide chain is written starting with the amino terminal residue. As you can see, the amino is towards the beginning side and carboxyl is towards the terminal side. Three amino acids can be joined by two peptide bonds to form a tripeptide. Similarly, amino acid can be linked to form tetrapeptides, pentapeptides, and so forth. When a few amino acids are joined in this fashion, the structure is called a oligopeptide. When many amino acids are joined, the product is called polypeptide, and each amino acid unit in a polypeptide is called a residue. Proteins may have thousands of amino acid residue. Although the term protein and polypeptide are used interchangeably, molecules referred to as polypeptide generally have molecular weight below 10,000, and those called proteins have higher molecular weight. Some protein consist of a single polypeptide chain, but others are formed of multiple subunit proteins, have two or more polypeptides associated covalently. The individual polypeptide chains in a multi-subunit protein may be identical or different. If at least two are identical, the protein is said to be oligomeric, and the identical units are referred to as protomers. The peptide bond is essentially planar. Thus, for a pair of amino acid linked by a peptide bond, 6 atoms lie in the same plane. The alpha carbon atom, the CO group from the first amino acid, NH group, and alpha carbon atom from the second amino acid. The peptide bond has considerable double bond character which prevents rotation about this bond. The double bond character is also expressed in the length of the bond between the CO and NH group. The CN distance in a peptide bond is typically 1.32 angstrom, which is between the expected values for a CN single bond and a CN double bond. Two configurations are possible for a planar peptide bond. In the trans configuration, the two alpha carbon atoms are on the opposite side of the peptide bond. In the cis configuration, these groups are on the same side of the peptide bond. Almost all the peptide bonds in a protein are trans. This preference for trans over cis can be explained by the fact that steric clashes between groups attached to alpha carbon atoms hinder formation of the cis form. The most common cis peptide bonds are X-pro linkages. Such bonds show less preference for the trans configuration because the nitrogen of the proline is bonded to the two tetrahedral carbon atoms, limiting the steric differences between the cis and trans forms. In contrast with the peptide bond, the bond between amino group and the alpha carbon atom and between the alpha carbon atom and the carbonyl group are pure single bonds. The two adjacent rigid peptide units may rotate about these bonds, taking on various orientation. This freedom of rotation about two bonds of each amino acid allow protein to fold in many different ways. The rotation about these bonds can be specified by dihedral angles. The angle of rotation about the bond between nitrogen and alpha carbon atom is called phi, and the angle of rotation about the bond between alpha carbon and the carbonyl carbon atom is called psi. The phi and psi angles determine the path of the polypeptide chain. A measure of rotation about a bond usually taken to lie between -180 and +180 degree. Dihedral angles are also called torsion angles. Now our query is, are all combination of phi and psi possible? G. N. Ramachandra recognized that many combinations are forbidden because of steric collision between atoms. The light values can be visualized on a two-dimensional plot called a Ramachandran diagram. 3/4 of the possible phi-psi combination are excluded simply by local steric clashes. Steric exclusion, the fact that two atoms cannot lie in the same plane at the same time, can be the powerful organizing principle, as clear from the diagram. Thank you.